Welcome back, everybody. We're going to start our last and final session on forensic biology and DNA. And to start us off, we've got Erin Butler here from the New York OCME office, and she's going to talk to us today about confirmatory proteomics for body fluid ID. Welcome, Erin.
Hi, good afternoon. I'm Erin. I'm from the Office of the Chief Medical Examiner in New York City, and I'm going to tell you a little bit about our work on validation of our proteomic mass spectrometry assay for identification of body fluids. So a bit of background, why do we care about identifying body fluids? You test a sample for DNA, you can find out the individual source of that sample, but that doesn't tell you the tissue-specific source. Your DNA is the same in every cell in your body. That information can be important in forensics. If, for example, you have a sexual assault case, knowing that DNA evidence that you've got a match to came from the semen stain as opposed to, say, shed skin cells, that is important information for that case, right? So in our assay, we're using proteins to identify body fluids, specifically blood, semen, and saliva. And we use proteins because proteins are differentially expressed in different tissues based on the function of those tissues. They're present in abundance, so you can identify them without amplification. And they're also relatively stable. They're going to stick around longer and through harsher conditions than DNA and RNA typically do. And protein identification with mass spectrometry is confirmatory. We're measuring those proteins directly with the mass spec. These are the specific proteins we're looking for identifying blood, semen, and saliva. These proteins have tissue-specific functions in blood, semen, and saliva, so they're present in high quantities, and they're also specific for those body fluids. These are the peptide markers that we're using to identify each of those proteins. Peptides are just short pieces of a protein. They're what results when you digest a protein, which is what you need to do to put it in a mass spec. And for the most part, we're looking for 3 peptides per protein. The exceptions are a couple of saliva proteins that are very short, so we only get 1 peptide for each of those. The specific mass spec method we're using is called multiple reaction monitoring, or MRM. It requires that our target peptides are defined and well characterized in advance. It uses triple quadrupole mass spectrometer. That's a picture of the specific triple quad that's in our lab. It's a SciEx 6500. And just real quickly, the way that instrument measures the proteins or the peptide mixtures that enter into the instrument. First, it selects and isolates the specific peptide based on mass. Then it fragments that peptide, and then those specific fragment ions are measured. So for our assay, we're targeting 4 fragment ions per peptide. And just very quickly, sample preparation is pretty typical of a proteomic mass spectrometry experiment. We solubilize protein, total protein quant, reduction and alkylation of that protein mixture, then digestion with trypsin, which results in a complex peptide mixture, those short sequences. And then that is separated by HPLC, which then goes directly into the mass spec. And this is what the data looks like when it comes off of the mass spec. This is a single peptide, in this case, a hemoglobin peptide. And those 4 different colored peaks stacked on top of each other are the fragment ions that we target. And then you also get information about retention time as well as the peak intensity or peak area. And so the way that we use that information to say, whether we've got the specific peptide we're looking for in the sample, we calculate a fragment ion score, and so that's just going to tell you whether those 4 peaks match what we expect from the peptide we're looking for. So for hemoglobin, this particular peptide, if the peaks don't come off in that order and in those peak areas in relationship to each other, the fragment ion score is going to be very bad. We're going to say that peptide's not there. But if it is there, it's going to look exactly like that, and the score is going to reflect that. The other bit of information we use is retention time. All the fragment ions should be eluting at the same retention time if they come from the same peptide. And then we also look at total peak area in order to determine if the peptide is present in the sample. And all of those metrics that we use for identification are generated per peptide based on experimental data. So the thresholds that need to be met for each of those metrics come from lots and lots of known samples. So once you've got your different peptides identified, how do we use that to say whether or not we've got the body fluid in question? For blood, we're going to require all 9 of the peptides we're targeting to be present in order to say we've got blood in the sample. For semen and saliva, it's a little bit different just because those body fluids are much more variable, individual to individual. The total protein that you get and the proteins relative to each other tend to vary a lot, and so our requirements for identification do reflect that. And so we're looking at a minimum of 7 marker peptides for both, plus some specific requirements for how many peptides need to be present from each protein. And that still gives us plenty of information to say for sure that we've got that body fluid in question. So here's our validation testing. This is the total summary of samples we tested in order to show that the process I just outlined does in fact identify blood, semen, and saliva where we expect it to. So of course we have lots of samples of blood, semen, and saliva. We have some mixtures. We have some non-human blood, semen, and saliva. Since this is a human-specific assay, we wanted to verify that specificity. And we also tested vaginal fluid and menstrual blood, again, just to confirm that we weren't getting false positives where we didn't want them. So first off, we tested a lot of just neat body fluid samples, neat meaning that we solubilized in a volume aliquot of liquid body fluid sample and processed it. And so those samples came from between 20 and 27 different individuals. And then each of those were extracted multiple times by different analysts. So we're getting a full characterization of what the assay can do. And those neat samples, for blood and saliva, we detected blood and saliva in those samples 100% of the time. For semen, there were 2 samples where we came up just short of a full panel of semen peptides, those panel requirements that I described in the earlier slide. So that gives you about 97% sensitivity there. And it turns out that those 2 samples both came from the same individual. And something we learned over the course of this validation is that semen in particular is hugely variable in the amount of protein that you will get from different individuals. So this individual happened to have very low total protein amount. And so that's likely contributed to the lack of sensitivity there. But other extractions of that same individual donor were also able to identify a full panel. So you just see some more variability of detection at the lower total amount of protein. So something else we did is we created some mock type samples, first using a known standardized amount of proteins. So the way we did that is we used those neat samples that had been solubilized and quanted, and then we prepared swabs using specific amounts of protein based on those total protein quants. And the reason we did that is because the different body fluids that we're testing for have very, very different concentrations of total protein per body fluid. So these numbers that you see in the table here are generated from the neat samples. So this is the amount of protein we get using our extraction method per volume of the original neat sample. And so you can see there's huge differences between the amount of protein concentration per volume of blood versus semen versus saliva. And I also want to highlight the coefficient of variation here. Blood is very low. You don't see a lot of variation in blood. It's very tightly controlled. Versus semen and saliva, hugely higher variation from person to person, so any individual sample can give you a wide range of total protein amounts, especially for semen. And so then these volumes listed here are just generated based on those averages, just to give kind of a ballpark of the volume of body fluid in a stain or a swab or whatever you would need to generate that amount of total protein. So you're still looking at pretty small amounts, even though it varies a lot per body fluid. And this is the result of those swabs with the standardized amounts of protein added. So this table, the colors represent the percentage of the samples that we were able to get a full panel of peptides, so an identified result from each amount of protein per each body fluid. So the light yellow is 100% detection, that sort of darker pink color is 0 samples detected. And so you can see based on this, semen has the best detection. We're getting full detection of all samples all the way down to 0.25 micrograms. Blood and saliva, not quite so sensitive. So we're starting to reduce the sensitivity at about 1 microgram of total protein. But again, the volumes we're looking at of blood and semen and saliva at 1 microgram, we're still in the nanoliter range, so pretty small. And then again, we also tested some less controlled samples with unknown amounts of protein. These are samples we got from our DNA QA/QC folks. They're used for DNA proficiency testing and are prepared with blood, semen, and saliva. So we're using them to see how well we can detect our body fluids on these somewhat less controlled samples. So we have some blood cards, we have blood and blood semen mixture stains on fabric, we've got saliva swabs and semen swabs. And for detection of blood, the samples that contain blood, we got all of the cards. Sensitivity on those was 100%. Fabric also 100%. For the blood stains, for blood and semen mixtures, we had 7 of those, and we got full detection for 6 out of 7. One of them didn't quite get a full panel. We're missing a peptide, so the sensitivity there is about 85%. 97% sensitivity for all those blood samples taken together. And then again, detection of semen, so those same 7 blood and semen mixture stains, when tested for semen, we identified semen in all of them, and then we had some swabs. We got 11 out of 13 of the swabs, so again, missing 1 peptide from a couple of them, so it's not quite full detection. And altogether, it's about 90% detection there. And saliva, 10 swabs, we detected saliva in all of them. We also did some tests on condoms. We just wanted to be sure that lubricant or spermicide that may be present on condoms didn't interfere with detection of semen. And based on these tests, it does not. We were able to detect semen under all these conditions, no problem. And we also did a small set of mixtures in addition to those blood and semen stains. And so this just basically functions as a kind of proof of principle for the mixtures, just to say that the presence of another body fluid in and of itself does not interfere with detection of other components of the sample. So the sensitivity is always going to be a little less because your total protein for any given body fluid is only a part of the total protein present in the sample. But if the peptides from the body fluid are there, we're going to get them. We're going to be able to identify it. They're there in amounts that above our limit of detection. So for these, for semen and saliva, all of the mixtures that contain semen and saliva, we identified all of them. And again, blood, for 1 sample, we didn't get quite a full panel. And so you see over several of these experiments I've outlined, blood seems like it's a little bit less sensitive than the others, which may strike you as counterintuitive given how much protein is present in blood, but the issue is really the dynamic range of blood. Of our 3 protein markers, we've got 2 hemoglobin markers that are present in just huge amounts, and our 3rd marker is a membrane protein that's going to be much lower relative to that hemoglobin, and so that affects the sensitivity of blood more than semen and saliva, where our markers are going to be generally present in more equal amounts within semen and saliva. So we also tested menstrual blood and vaginal fluid, just again to confirm a lack of any false positives. So of course, blood was detected in menstrual blood, but we didn't get a false positive for semen and saliva. Same with vaginal fluid, no detection of any of our body fluids. And then similarly, our non-human samples, so the samples that we were able to test, mostly pets and agricultural samples. We were also able to get a gorilla blood sample, so that was nice. We were able to test a close relative of humans. But in all cases, even gorilla, you can see that column in the middle, it says detected, everything is 0. We didn't get any false positives from non-human samples. And just as a quick explanation of that, we designed those protein panels to be human-specific based on a sequence search from the NCBI protein library of all available protein sequences. So as thoroughly as we could, we made sure that no other animal would get a full panel of peptides, even though there's going to be a lot of homology for some sequences, just because we've got proteins that are, have very important functions in the body. So for example, cat has 4 blood peptides shared with human, and we identified the 4 that we expected to identify, but not the other 5. So of course, that's a negative result. Same with gorilla. Close relative, 8 out of 9 are shared with human. We did identify those 8, but that 9th was missing. Again, consistently, multiple extractions. That peptide was never present, which was the result we expected there. And we were also able to confirm that vasectomy doesn't affect the detection of semen. 7 of our semen donors happened to have had vasectomies. We had no problem identifying their semen. And so finally, everything I've been showing you so far is related to sensitivity of the assays, showing you how often we can detect blood, semen, and saliva under different conditions. But in forensics, possibly the most important thing we want to know is, you test an unknown sample, says it has semen. How sure are we that that's true? What's your confidence in that positive result? There's a couple of ways to get at that statistically. One of them is specificity. So specificity is for a binary test. Binary just meaning, our test for blood, you've got 2 possible results. Blood is detected, blood is not detected. So for a test like that, specificity is the number of true negatives divided by the total negative samples. And so in this case, negative samples means everything we tested that is not the body fluid. So blood negative samples is all non-blood samples, semen, saliva, negative controls, all the non-human stuff, and so on. So that's a high number of samples. So our confidence in that 100% specificity, 100% meaning we had 0 false positives throughout all of this testing, and the lower bound of that confidence interval was quite high because we're taking that out of, between 400 and 500 samples in all these cases. So it's between 98 and 99%. Another way of looking at that is precision. So precision, again, for a binary test is the number of true positives over total positives results. So it's focused on positive results rather than negative samples. So just a slightly way of getting at more or less the same information. But again, in this testing, we got 0 false positives. So the total positive results is equal to the total positive samples tested, and the lower bound of a 95% confidence interval on that is between 95 and 98% for all of our, our 3 body fluids. And finally, including the false discovery rate, this is just the inverse of precision. It's a little bit more intuitive to talk about. You say your false discovery rate is 0%. That's a fairly easy to interpret statement. And the upper bound of that 95% confidence interval is just going to be the inverse of the one on precision. So between 2 and 5% for each body fluid. So all that to say, essentially, the specificity, the confidence in a positive result of this assay is very high. If we test something, we say it has blood in it, we can be very sure, based on these results, that it's definitely blood. And that's all I've got for you. I just want to thank everybody in OCME proteomics lab who ran all those samples. And of course, I want to thank NIJ for funding this work, for the validation as well as the research and development that preceded it. And the OCME lab director would like you to know that we are hiring, so you can come talk to us during the conference or just go straight to nyc.gov/jobs and apply. Thank you.
Thank you for the great talk. It was very informative. I have a question regarding the way you were detecting the peptide. Okay, so were you looking at the variant peptide also? Variant peptide combined with the non-variant peptide for the body fluid.
In this case, no. There aren't any variants. We checked available variant databases for our peptides. There aren't any variants with high frequency enough to be worth testing for, except for one of the semen-Magellan peptides, which you do see some people who are heterozygous for, which probably may affect our sensitivity detection of semen in a small portion of the population. And so there's a possibility that in future versions of this assay we might try to add the variant as well. But for now, no.
Okay, thank you.
Thank you so much for the talk. I just have a curiosity question. You probably have mentioned it. How was the saliva collected? Was it a spit or a buccal swab or?
Both.
Both.
Yeah. Some of the swabs, the ones that were the PT tests for DNA were buccal swabs, but we also tested just neat saliva and those swabs that were prepared by depositing liquid saliva on a swab.
Okay, so there wasn't any difference between whether it was a buccal or a spit for the saliva?
Not in what we tested, no.
All right, thank you.
We have at least one online question. I think you already addressed this a little bit. If you only detect some but not all of the target peptide regions, how do you interpret that result?
So I don't know if I can go back to the slide. So the specific peptides that we require to be present in order to say that the body fluid is there varies a little bit for our different body fluids. We have a defined panel. If what we see in the sample doesn't meet those requirements, but it's close to it, basically if we get more than 4 peptides but not a full result based on the definition I showed you, we call it inconclusive. So that's to say that we have some evidence that the body fluid is there, but we can't, but not quite enough to say with certainty. Less than that, it's not detected. Oh, there it is.
Okay, sorry, I have to translate the question first. 1 second. Did you test with the with the semen detector, did you test in samples of decomposing dead bodies?
No, that's not something we tested so far.
Similarly, there were a couple different questions on how does the body fluid ID method perform with degraded body fluid samples? You already addressed mixtures. And how would degradation of the proteins over time affect the detection? Would time be a factor in which samples you choose to test with this method?
We are definitely planning to do more extensive testing for time and degradation, but I can say that those proficiency test samples some of them were as old as 10 years old for the blood cards. So those were all between 1 year and 10 years. And it didn't have effect on sensitivity. So the places where the sensitivity was a little lower for those samples were not the oldest ones.
Do you have any plans to investigate the effect of ALS and other visual/chemical scene detection methods on the ability to successfully ID body fluids with this method?
I'm sorry, of what?
The effect of ALS and other visual/chemical scene detection, alternate light source. Sorry, I could.
Yes. We tested a couple of them early on in development. That was a while ago now, but they didn't have an effect. Yeah.
Okay. One more online. What is your extraction procedure for hemoglobin?
The sample prep method was up on there. I kind of breezed through it because I know I wasn't going to have a lot of time to get to the results. But our extraction buffer is, let's see. Oh, can we go back to that slide? No. A little further.
Further forward.
Further down, yeah. There we go, this. ABC with SCC. That's not the strongest buffer you could use, but it's the one that requires the least amount of cleanup in order to put it on our mass spec. So for time, cost, and throughput reasons, that's what we use. I think that's hopefully answers the question.
Yes, thank you, Erin. And next we have Tracy Dawson-Green from UVA. And sorry, no, it was a test. VCU. She's going to be talking to us about sample prep and prediction models for early mixture detection.
Tricky. Sorry, that was we don't like that around Richmond. We don't like to get mixed up with the wahoos. All right, so I'm going to talk a little bit about our work today. I don't know how this works, this thing, Tracy. Hit the green button. Is that what I do? Is there a pointer? Yes, okay, got it. All right. So before I get started, I just want to go out right away and say that the data I'm going to present today represents the work of several graduate students, previous and current, and I will recognize them individually at the end of the presentation. So jumping right in, as probably everybody in this room already well knows, currently we are unable to detect whether a sample is a mixture or a single source sample until the very end of our forensic DNA workflow. And this shortcoming, I would say, really exacerbates the problems that we already see inherent with mixtures and low-level DNA samples. And so we, in talking about this, we started thinking it would be very nice to have this information a little bit earlier on in the workflow, especially if we could have it before STR amplification. Having more information earlier on, say at the quantification stage, can certainly help you triage your samples, make some protocol modifications that may help to mitigate the challenges that we see with these types of samples. For example, if you have a low-level sample or touch item, a small evidence item, and you have multiple swabs from that evidence item, and they all go through their own extraction process, and you end up with 3 unique DNA extracted DNA samples. If you could know at the stage of quantification that they are all single source, and particularly if you could know they're of the same type, you could potentially more confidently combine those extracts so as to avoid the limitations associated with low-level DNA interpretation. Alternately, if you knew that a biological evidence sample was a mixture at that stage, you could also go ahead and adjust your STR input so that you could try to avoid dropout of minor contributors in that sample. So we started thinking about this actually when I was up at the ATFE lab talking with Todd Bill, and he was talking about how useful this type of assay would be. And his initial, in brainstorming, his initial thought was, hey, let's just add in some D1S80 primers, which most of you in this room probably have no clue what that is, but long before I entered the field, we used, that's a joke, we used to use D1S80. It's a little bit longer than our STR loci though, and so we started playing around with that, broke out the primers from the back of the freezer, and adding those to our quantification assay, busting through the seal of the quantification plate, and then looking at those products on a quick flash gel. We were able to see mixtures, as you can see there with the three bands showing multiple or more than one contributor. However, we had some resolution problems because those alleles can be fairly large. We moved on to triallelic SNPs, which was pretty cool. We got that to work, but only when we used fluorescent scorpion primers for this. And then we were using so many channels of our qPCR instruments, it was going to be impossible to incorporate that particular approach into a quantification assay. So then we moved on to STR targets, and at this time, Eric Buell from the Vermont State Police had a couple of papers published where he was looking at genotyping STR loci from melt curve analysis using the qPCR instruments. And so we thought, okay, well, let's take a look at this as an approach. And we knew STRs would be easier to incorporate into our quantification assays because they're of the right size. And we knew if we picked the right targets, we could potentially duplex them and detect them in a single channel of our qPCR instrument. So that's what we went with, aiming for a melt curve analysis that could be added on the back end of the qPCR amplification. So just in case you're not familiar, melt curves can be used to look at amplicon variations, particularly variations in sequence and amplicon size. You would expect shifting of the primary melt temperature when you have those differences. And usually the raw melt curve data is shown there on the right. I think, can I? Oh no, that's not right. Oh, there we are. So this is raw melt curve data, and typically we incorporate a double-stranded DNA binding dye or an intercalator dye, and then as the temperature is increased over a range of temperatures, slow incremental increases will result in denaturation of that double-stranded DNA, and once you get to about 50% of that double-stranded DNA denatured, then that is the primary melt temperature, and that's usually shown in the derivative plot like is shown here. So for a single amplicon, you can see a nice single melt peak there, and that's the primary melt temperature. So this should shift with variations in amplicon size and also sequence. So that's kind of the basis for what we were going for here. We know that melt curve analysis is available on most modern qPCR instruments, including ones that we have in forensic labs, although some are higher resolution than the others. And so we started thinking about this using those two instruments, specifically the ABI 7500 and the Rotor Gene Q, which is a higher resolution melt curve with a dedicated channel for melt curve analysis. So we kind of took it in that direction and started playing around just to see if we could actually genotype STR loci using this approach. And we did see shifts in melt temperature with different genotypes, but we also saw some closely related genotypes that kind of grouped together and didn't have much of a shift. So when we looked at mixtures using this assay, we also noticed a temperature shift. So when we combined two individual contributors of two different genotypes, we saw a shift, but we also saw a change in curve morphology, which was pretty interesting. And we knew that to kind of get a handle on that, we were going to have to use something, a method that was a little bit more quantitative than just looking at these curves. It wasn't enough for us to be able to distinguish between mixtures and single source just by looking at the resulting curves. So we went over, what we do, we go over to the Department of Statistical Sciences and Operations Research and we say, hey, help, we're not sure what to do. And that was a great move because they had a lot of ideas about different prediction modeling, prediction algorithms that we could use to analyze data from these melt curves to be able to determine whether a sample was something about the contributor status of a sample. And when we use these types of prediction modeling approaches, you first have to develop a set of standards that will serve as standards to your training set that will train the software on the different genotypes, in this case, that we were looking at. So we were included in our initial study 7 different samples from 7 different genotypes across those 2 different STR loci, D5 and D18, D5 and D18. So we took those samples, we ran this melt analysis just straight out using the amplification and then the melt on the back end, and imported those entire melt curve data, which are very large data sets, particularly within the higher resolution instruments, and then we imported them into other software where we could use them to generate splines. Splines are kind of mean melt curves that were developed for every single genotype of each locus that we were analyzing, and then the spline coefficients are then used to actually help to compare to unknown samples to make predictions about those unknown samples. So we used initially R software as well as QIAGEN's ScreenClust software, which is a high-resolution melt curve analysis software. And all of the other unknowns that we had, those are in our validation set. So the terminology is irrelevant except to say we tested the trained models using unknown samples, and then those software, they spit out these confusion matrices like are shown here, where you have known genotypes going down this way, and you have predicted genotype groups here, and then where the samples are predicted are all throughout here, and those on the diagonal would represent the samples which have predicted accurately. And so when you look at this example of this data, what we were consistently seeing initially was that the LDA, the linear discriminant analysis, that model was working better, which we were using in R, than the PCA type of analysis. And so we kind of discontinued our work with the PCA and focused in on LDA, and specifically looking at some other machine learning tools in addition to LDA that might be more appropriate for this type of data. So we looked at a couple different SVM tools, as are shown here, as well as LDA, and our initial attempts at genotyping those 2 STR loci using this prediction modeling scheme on 2 different instruments with HRM analysis, and you can see some of that early data we got here. And what we took away from this data is that, number 1, the SVM models do work a lot better, and number 2, the ABI 7500 pretty much syncs for melt curve analysis, which we knew because it's a low resolution. It's not built as a high resolution melt curve analysis. So we wanted to move on and really think about, with this grant, how to expand our investigation of those machine learning tools, and also we wanted to really use those to evaluate our original question, which, if you remember, was actually, can we use this data to predict the nature of a sample as that of being either from a single contributor or from multiple contributors? And we wanted to do that inside of a commercial quantification assay, because we don't want a separate assay. So this was a pretty lofty goal, and you know that when you go screwing around with these proprietary kits, you can find some surprises along the way, and you have to be really careful. So the first thing we wanted to do was to see if the chemistry that we were altering was going to affect our predicted melt curves. By this time, we had characterized melt curves from these loci quite a bit, and we wanted to know if this was going to alter it. So we incorporated this assay in the QIAGEN Quantiplex kit. And as you can see, the Quantiplex kit, when you add a melt stage on the back end of the amp, and you add an intercalator dye, like in this case, Eva Green, the kit itself produces no melt product. So we knew we weren't going to have interference with our expected STR melt products when we incorporated into this kit. We also had to look very carefully at quantification. After all, that's why that's the primary use of these kits. And when we looked at quantification, we saw, with this modified chemistry that had this melt curve on the back, we saw really no significant changes in the standard curve. The R-squares, y-intercepts were all within standard ranges. The slopes did decrease just a little bit, but by about 2 cycles, but every single standard in the curve was decreased by the same amount, so we didn't expect that to have an impact on our quantification data. And in fact, when we looked at the quantification data compared to the same samples tested with a standard quantiplex assay right out of the box using manufacturer's recommendation, that data was actually tighter than inter-run duplicates tested using that commercial kit. So we thought, okay, the quants are good. And now by this time, my colleague Sarah Williams, she had acquired a new qPCR instrument that also had high resolution melt curve capabilities. So we thought, okay, perfect. We're going to ditch the 7500 and use the Quant Studio. The Quant Studio, there's a couple models, 5 and 6. They're both now new. They're newer, they're better, they're faster. If you have an opportunity to replace your 7500, jump on them. They have added capabilities, tighter control, better reproducibility, 5 color channels, the opportunity to incorporate custom dyes, and high resolution melt curve capability. So we also brought in this instrument on this project and started working within this platform. And we evaluated this, this STR analysis that's integrated into the Quantiplex kit with an HRM function on the back. We started looking at this on this platform to kind of determine to look at the single source genotyping just so that we could determine the best prediction models. And this is the same table I showed you before, but this time with this Quant Studio added in. What we were surprised at the low single source genotyping prediction accuracies, but we were able to determine the best performing machine learning models to move forward with for our actual question, which is whether or not this approach could be used for single source and mixture discrimination. So this is that data. Again, thanks Tracy, the only difference is here is that samples which predict as samples that predict as a single source anywhere as a single source are considered single source at that locus. Samples that predict as a mixture are called mixtures at that locus. And then having the two loci combined into this assay allowed us to be very conservative about those calls. And any single sample that called as a mixture at either locus would be a mixture. And that's how we approach this data. And this is the effectiveness of that tool. Around 90% of the time, we can predict in the Quantiplex kit at the same step, using the same DNA sample you're quantifying, whether or not your sample is a single source or a mixed sample. But I know what you're all thinking. Who uses Quantiplex? Nobody. So we knew to make this more viable for the practitioner, we were going to have to move into a different kit. So we started working in the Quant Trio kit, and most of you are more familiar with this kit. And so I won't go through the details except to tell you that it's a huge pain because there's a bunch of stuff going on there. There's a lot of targets. Every single channel is utilized. And also it's a TACMAN assay. So guess what? It does have double-stranded product at the end. And sure enough, there it is, the orange line. The kit just with the dye, the HRM dye alone is throwing off a melt curve product. And when you know it, it's right in the range we don't want it to be in. But we move forward nonetheless. And I'm going to skip a few of these slides on the development of this. We had to do a lot more optimization with this particular assay. We had to find a new dye that wouldn't overlap so much with any of the quant targets. And so we settled on the Cyto dyes. We tested a few of those and ended up with the Cyto 64 dye because it only overlaps with the June dye that's on the IPC target. Anytime you screw around with that, you're going to have some shifts in your standard curve, like I mentioned before. So we also had to adjust the analysis settings. And when we did, we had standard curves that looked good. This time, our slopes were slightly lower, but they were consistently lower. And so that is okay in terms of quant. Our Ys, our R squares were all within normal values. Even our IPC was unaffected. And I'm going to skip these couple of slides just to say, to show you that the quants were again very similar to what we saw with the Quantiplex kit, in that they were essentially there's no more variation in these quants compared to the regular protocol than the regular protocol had itself over multiple runs. So we were very satisfied with the quant, and the resulting SCR profiles also looked really good. Oh wait, sorry. The resulting SDR profiles looked really good in terms of the profile quality. Of course, there's other targets in there, so we had to make sure that they weren't affected. And again, we saw no change in the degradation index, the male-female ratio. The IPC flag for humic acid the detection was the same in our modified chemistry. Hemotin was a little bit delayed in our modified chemistry, so this seems like it's going to be a little bit inhibitor dependent. We may need to test more. At the end of the day, the whole question with this is whether or not we can distinguish between single source and mixture samples, and about 80% of the time we got that right with this modified quant trio assay, so we were happy with that. We're moved on now to do some additional developmental validation studies. And we did notice a little bit lower inter-run prediction accuracy reproducibility when we did these studies, and we later kind of found out that, oh, that has to do with different kit lots. So it turns out that when we test with different trio lots, it still works, and it works really well, but this suggests we have to potentially rerun the training set every time we get a new kit lot. And that's not practical and isn't going to work. So we're working on right now building consensus splines using our data for each genotype across all the kit lots that we have, and we're hoping that will remediate that issue. The other studies that we have moving forward now, we're just analyzing data from these studies. Most importantly are looking at, I failed to tell you that all the mixtures we looked at so far in the development of this assay were 1-to-1 two-person mixtures, which is obviously the best case scenario. So we realized that we have to expand our studies to look at mixtures with more than 2 individuals and also across a range of ratios. And lastly, remember that we're only using the 7 most common genotypes in our training set here. We've limited our initial proof of concept studies to that. So we have to expand our training set for this to be applicable in a casework setting. And we've been doing that using synthetic data with our statistician. And I know that's a very scary word, but it's really not a fully synthetic data set. It's a hybrid training set that includes real training data, but then for the genotypes that we don't see in our population samples, we use within the software polynomial regression splines as well as interpolation to get data for the genotypes that are missing from our real sample sets. And with that, we have these hybrid synthetic data sets, and that's what we're working on right now. We're having a little bit of a mixed bag with those, but we're seeing more positiveness now in that approach. It's really just about trying the right algorithms to get this to work. That's what statisticians do. They keep trying things until something works, and that's what we're doing. So I guess in conclusion, that's kind of where we stand with this project. We have this assay functioning within 2 different commercial kits. We are able to determine pretty consistently and with high accuracy rates whether a sample is single source or mixture at the quantification stage. And we're able to do so without impacting the kit's ability to accurately quantify human DNA. And with the trio specifically, we also are able to do that without altering the other supplemental information that you get from that kit. So we're completing these validation studies, working on this synthetic data set, and we're also in the process of building a user-friendly interface that could be used by the end user that would allow easy import of melt curve data, and then it would pop out a report just with the prediction of each sample that was tested. So that's kind of where we stand. And as promised, I want to point out the students whose work I've been discussing today. My current PhD student, Chasten Smith, Dia Torres, who is from the FBI now and is here. She probably could have given this talk better than me. Dre Williams, who's at PG Crime Lab, and Hannah Wines in Houston, Darian at PPD, Christy Kuhner at Bodie, my former lab manager, Jordan Cox, who's now at Thermo Fisher, of course, our ATFE lab partners, Todd, Steve, and Caitlin, and most importantly, the NIJ for funding this very important work. Thank you. They're stumped. Good, let's go.
All right. Thank you so much for sharing that work with us. I love this project. I'm in love with this project. I'm over here. Hi.
Oh, okay. Oh, good. Thank you.
Yeah, it's really, really great. So I'm going to apologize in advance because I might ask a statistical question. But
Uh-oh.
I
Okay.
was kind of shocked that PCA didn't work as you were leading up to your slides. I was really anticipating that to be the punchline.
Yeah.
And when it wasn't, I was like, what happened? Like, what did you notice any trends in the data that?
Yeah, well, I didn't, except that it failed. But what my stats, it wasn't just very good. Also, the ScreenClus software is really clunky and difficult to use. You have to run the standards every time on the play stuff, so we didn't really like the software too much. But the PCA, I later learned, is not really good for these types of very large data sets with machine learning. It's not as good as some of the newer machine learning algorithms that have been described. And so I don't know exactly why, except to tell you that to our statistician, it wasn't very surprising. I think PCA looks at the differences more within sample groups instead of the differences between sample groups like a LDA or an SVM might, and that just tends to work better with this type of data. These are very large data sets. We have like over 1,000 data points on the Rotogene Q and over 2,000 data points exported for every single melt sample from the Quant Studio. So they're very large data and just does better with these machine learning tools.
Thank you.
Mm-hmm.
All right. Thank you so much. I have just two quick questions. When you said something about the accuracy being 80%, do you contribute the 20% being some mixtures of contributors who have like shared alleles probably? That's why there's no...
Yeah, more likely those, and I think we actually went back, Dia may correct me if I'm wrong, and we do look at the ones that are inaccurately typed. We do look at those carefully, and usually they're closely related genotypes. So like an 1111 or an 1112, 1113 or something like that, that just is beyond the resolution capability of the instrument. I mean, there's no melt curve instrument right now, I think, in the world that will really be able to get it right every single time. But we look at this as supplemental information, and actually Todd at the ATF lab, he said, I don't even care if it's better than half the time. It's useful for me in a casework setting. So it's not like you're writing reports based on this. It's really just another piece of information to guide your next step in the workflow. So we were thinking 80%, is pretty good, and we've been rolling with that. But we haven't ruled out the possibility of looking at other prediction modeling tools as well.
And the other quick question, for the training model, would you think you would need to do a different training model based on the different populations, or that wouldn't make a difference?
Yeah, I don't really think so, because the genotypes would be the same. So the most important thing is that we have a training set that encompasses most of the commonly observed genotypes, and we definitely want to have real data on both ends, on the low end and the high end, and we do. And then we're filling in with the synthetic data. So to us, I think that's the most important thing, because you're going to still see the same genotypes. There may be some slight sequence variation, though, between population that can contribute a little bit, but it shouldn't be, those shouldn't really contribute too much to the overall melt curve morphology.
All right, thank you.
Mm-hmm. Okay, thank you, Tracy.
Thank you, Tracy. All right, so we're going to try and stay a little bit more on time here going forward. That's my fault. I apologize. Next is Dr. Julie Valentine. She's going to be joining us. She is from Brigham Young University, and she's going to be discussing with us some findings from a very important study for male sexual assault victims and multidisciplinary implications.
Thank you, Tracy. And if you haven't been told this already, happy Valentine's Day. This is a big day for our family. We send out Valentine cards instead of Christmas cards, the whole bit. So happy Valentine's Day. I'm going to be talking to you about a side study that has developed out of a grant that we have received, and we're so appreciative of from the National Institute of Justice, which is actually looking at development of a machine learning model with sexual assault evidence from sexual assault kits. I will tell you, so far, our attempts to develop the machine learning model have not been hugely successful in that we found that we're not getting better results there than when we look at what's happening in the lab. But more to come on that. No conflict of interest to report, and here are my co-authors, and I also did not list, we have had an army of research assistants on this project. So as we were looking at factors or features associated with development of our outcome variable is development of a CODIS eligible, so STR DNA profile, we found immediately that gender had a huge role, so much so that we really could not analyze our female genitalia cases with male genitalia cases. So we are not looking at gender orientation. We categorize it as who has female genitalia and who has male genitalia. And we found huge differences. So now we have a side study that has developed about male sexual assault victims and the DNA analysis findings. So traditionally, as we know, many think of rape as female victim and male perpetrators, but we know that all genders are affected. There's a great website called oneinsix.org, if any of you are interested in looking at that, where it really talks about the larger numbers of male sexual assault victims. The challenge is, other research has shown, is that they are less likely to disclose. So our study methodology, we are a retrospective exploratory study. We code data from sexual assault medical forensic examination forms. We currently have, we've gone from 2010 to 2022. We have about 10,000 cases. We collect 241 variables from the sexual assault medical forensic exam forms, and then we follow those cases through to the kit submission and then the DNA analysis findings. So for this side study, we are looking at what differences exist between sexual assaults of male and female victims, and then how do DNA findings from male sexual assault victims compared to female, and the distribution of DNA profiles from swabs from male sexual assault victims. So this study that I'm sharing with you, or these numbers, are from 2010 to 2019. We are currently rerunning these same analyses because we have a larger N of males. But in this, we had 6,627 fully collected sexual assault kits. You see our number of those submitted is dramatically lower. I am happy to report that we did a large study on sexual assault kit submission, and those findings led to legislation that passed in Utah in 2017 mandating the submission and testing of all sexual assault kits. So we went from 36% of collected sexual assault kits in which victims said they wanted to prosecute their case and interview with law enforcement to 99%. So we are very excited about that. But our numbers, you can see 4,404 from female, 193 sexual assault kits from male victims. So I'm first going to share with you some features related to, not to DNA, but just to overall comparing male and female sexual assault victims. And as you can see, there was a statistically significant difference between the assailant relationship in male and female sexual assaults. First, strangers. We found that male victims are actually more likely to be sexually assaulted by what our category of stranger or our category of others. Let me define those for you. Stranger, for our research purposes, is defined as the victim did not know the perpetrator's name and did not have what we have called a meaningful interaction with them. and we categorize that about 2 hours. So, for example, if a victim says, I went to a party, I was really tired, I decided to go and to a bedroom and sleep, and I woke up and he was on top of me. I don't know his name. I saw him at the party. We'd categorize that as stranger. But if the victim said, I was at a party, I met this guy named Jim, we played some games together, we watched a movie together, and then later I got tired and I went to bed and I woke up and he was on top of me, we would categorize that as acquaintance, even though they had just met that night. Now, we categorize other, generally that's someone in a position of authority. So it's a boss, a teacher, bus driver, or a family member. So you can see on our male victims, we have quite a bit more stranger and other. We have less spouse, partner, but look at the ex-partner. When we started this study in 2011, we noticed that there were a number of sexual assaults by ex-partner, mainly ex-boyfriend. So we decided to code for that. And you can see on our males, we actually have more sexual assaults by ex-partner than current spouse partner. Then we looked at differences in demographic information. And we found that male victims were much more likely to have a physical or mental impairment. Physical or mental impairment such as cognitive delays, being blind, deaf, anything that impairs the senses. We have another study where we're looking at what are the vulnerabilities for sexual assault. You can see for the males, a huge vulnerability is a physical or mental impairment. We also found that males were more likely to have multiple suspects in the assault than females. Victim grabbed or held was less, but victim being hit was higher for males, and these are all statistically significant. Victim being strangled is also higher for our females, but higher use of weapon in the male sexual assaults. Female victims had more injuries documented than our male victims. So then when we look at the sexual assault features, you can see for our male victims, much more anal penetration with penis, finger, hand, mouth, or an object than the female. Also, more oral contact of the victim's genitalia on males. Condom use. You can see that number is low. The American College Health Association did a survey and asked college students how many used condoms. It's about 42% in consensual relationships. When I do training with law enforcement, I talk about this low condom use should send a bell off that that's a sign of a non-consensual sexual act. Because in consensual relationships, people generally talk about how are we going to prevent sexually transmitted infections, how are we going to prevent pregnancy with female victims. Low condom use in sexual assault cases. Lubrication use, much higher in the males. The number one lubrication, any guess? Spit, saliva, yep. And when my victims, I'm still a practicing forensic nurse, and when my patients tell me that, I'm immediately like, oh, that's gonna heighten my DNA yield, right? Ejaculation. Now this is if they said, yes, there was ejaculation. 26% of the males said yes, 33% of the females said yes. But the higher number is victims say, I don't know. 57% of our victims, when we combine male and females, when we ask about if there was ejaculation, they say, I don't know. Another sign of a non-consensual sexual act. They're not focused on that. In consensual sex, most people know if their partner ejaculated. We don't see that in the sexual assault cases. So then we looked at the DNA findings. So remember, our outcome variable is CODIS eligible DNA findings. When we looked at endus, we have variables where we look at estus and endus. This is just endus. 34% of victims with female genitalia developed profiles that were entered into endus, where males, we only had 19%. We have a publication that we have submitted that is just on the females and DNA. So when we look at features that affect the development of a CODIS-eligible profile, we find that age is statistically significant for both males and females, but in a totally different curve. Now that says an age of 10, that actually should be changed to 14. Our cases are adolescent and adult, so that's at age 14. But you can see the males, we see this sudden decline from 14, whereas the females, they will hold fairly steady until 30, and I don't have beyond 40. But what we've found with female victims is around age 50, when many females go through menopause, we see a huge decrease in the development of a CODIS-eligible profile. We think that drop in estrogen, which decreases vaginal lubrication, likely lends itself to a decreased yield of DNA. Here are non-significant features on development of CODIS-eligible profile, no huge surprise, race, victim-assailant relationship, strangulation, and documented injury. This is for males. For females, we found that strangulation and documented injury, both non-anogenital and anogenital, are statistically significant features in development of a CODIS-eligible profile. Time between assault and the examination, always a huge question. How far do we go from when a victim is raped to when we say, all right, it doesn't make sense to collect evidence anymore? We don't have very many studies that are actually looking at this in practice, and that's what our study is trying to do. So for males, development of a CODIS-eligible profile is a mean of 13 hours. No CODIS-eligible profile is a mean of 31 hours. For our females, we went on our study just looking at the female victims. We have one case where we developed a full STR-DNA profile 8 days from the time of the assault to the examination, and the victim showered twice. It was from vaginal swabs, 8 days. For our males, the longest we have gone where we have developed a full CODIS-eligible profile is 57 hours. It's about 2.5 days. Huge difference in that time frame. Significant feature on development of a CODIS-eligible profile, not a huge surprise, bathing or showering. That also is statistically significant for females. We find that females that bathed or showered, 21% develop a CODIS-eligible profile. If they don't bathe or shower, it's 42%. Multiple suspects, the old mixture, right, that we just talked about, make it more difficult to discern. So we find that those cases for both females and males where there's multiple suspects, less likely to develop a CODIS-eligible profile. And even though we don't see a lot of condom use, if there is a condom use, less likely to develop a CODIS-eligible profile. Increased association, if they say yes to ejaculation, more likely to develop a CODIS-eligible profile. Grabbing a victim and hitting a victim, because that is more contact, we see increased development of a CODIS-eligible profile. So for our males, we looked at which swabs developed the CODIS-eligible profile. I want you to think back that I shared that there was much more likely to have anal penetration with penis, finger, hand, or object. But isn't it interesting that those are not the swabs where we are developing those CODIS-eligible profiles, rather penis scrotum. is the number one. Now, they also have oral contact, so that's saliva, we think, or there may have been some ejaculation or just epithelial cells that developed that on the penis and scrotum. Body was even above rectal slightly. Oral, clothing, underwear, and condom retrieved at the scene. So, implications of findings. One that is not up there is we may want to really encourage male victims to have those exams really early on with that decreased time frame that we are seeing. Only up to 57 hours is our farthest that we have gone out. Males with physical and mental impairment, much heightened vulnerability for sexual assault. Again, the anal penetration is more frequent, but we have to make sure that we are collecting those swabs from the penis and scrotum. We found the highest yield there. We believe that the difference between the male and female being much more likely to develop a CODIS-eligible profile from a female really is due to the anatomical differences. That vaginal vault just holds on to that DNA more than a rectum holds on to the DNA. Further research, we are running these numbers again. We have now, I think it's 450 male cases, so we are re-looking at all of these findings and hoping to get these out published soon. There's our references. I can take questions.
We do. Does your data set include sexual assaults that occurred in jails or prisons?
It does. That's a small percentage, and to make sure that we stay with our de-identified data, we aren't sharing that, but it does.
Another question online, was there any significant difference between the length of time in developing CODIS-eligible profiles from anal swabs for male and female victims?
I have not looked at that specifically. We have looked at internal swabs, specifically vaginal swabs with females, and have seen that that's really, as time goes on, that's where we're developing the DNA profile. Everybody's like, we're almost done for the day. Thank you, Tracy.
Thank you, Dr. Valentine. All right, so we have our last presentation for the day. So this is NIJ Shade, and she is at the Rutgers University in Dr. Catherine Gerkachev's lab, currently earning her PhD, and she's going to be talking to us about single-cell strategies.
Thank you. Hello. Okay. Thank you so much, everyone, for staying for the talk. It's the last presentation. I'll try to be as informative and on time as possible. So my PhD project entails selectively analyzing and interpreting DNA for multiple donors with a full single-cell strategy. So even before I get into what my different steps were in my PhD, let me just walk you through the aims. So the overarching aim for my PhD is to develop and compute the weight of evidence for an admixture using a single-cell strategy. Sub-aims are to implement a suspect agonistic clustering method, and finally, to use this data that we get from clusters in a forensically relevant way. Before I get into the nitty-gritty details of my work, just let me take a step back and explain the difference between a bulk mixture and a single-cell pipeline as to what actually happens in the lab and how do we process the data so that we can work with single-cell. So traditionally, what we do is, so when we if we go to a crime scene and we get a sample, the first step is to take a small portion of that sample, and that sample goes to extraction. So in the extraction phase, we get the DNA out of the cells, which can be shown right here. And we get the DNA molecules out, and after the extraction, we go into amplification. Now, I'm going to slow down here and explain as to what happens exactly here in the bulk pipeline. When we go from extraction to amplification, we take a small aliquot from the extraction tube that goes into amplification, leading to, we tend to lose a few DNA molecules, which can be indicated here. The light green ones and the light blue ones were not picked up when the aliquot was taken from the extraction to the amplification phase. Now, this is just a demo to show as to what happens process-related problems. And this can be depicted in the final step through the electropherograms, where we can see that 16, 17 alleles were not detected, leading to loss of information. Now, this is very, very vital because, as we all know, that when we have a mixture sample and when we are determining the number of contributors, the less information we have in the electropherogram leads to erroneous results in the number of contributors. So what our lab is doing is trying to work through this problem, and that's where the single-cell pipeline comes in. So what happens here is, with the sample that we had from the crime scene, what we do is we sequester the cells into each tubes. In that tube itself, we add in the extraction mix, and the amplification mix as well goes in the same tube. That leads to a direct-to-PCR extraction and then the PCR amplification, leading to no aliquoting step. So the ultimate, hypothetically, we would see that we would get an electropherogram per cell with near to full complete profiles. Now, this seems to work theoretically, like I explained, but does it really work in the process, and can we actually sequester the cells? So now that's where my PhD comes in. So the first step, obviously, is to, when we are working with direct-to-PCR and we are trying to add in the extraction and the amplification mix in the same tube, is to check the compatibility between the reagents. So the first thing we did was we published a paper recently where we looked at 4 direct-to-PCR extraction methods that are readily available for forensic science. We looked at Viagen direct PCR solution kit. We looked at forensic gem, DePere, and PicoPure extraction. So in our lab, we work with Global Filer, and we use 3500 for CE. So we were developing a method where we have all these extraction kits compatible with our Global Filer and see what works best in our lab. So we looked at 3 different matrices to evaluate the EPG quality. The first we looked at was frequency of alleles detected, the beta value, which is the sloping effect you would see with increase in the molecular weight if there is decrease in the peak height. And we also looked at the average peak heights per cell. Looking at the data through the 4 direct-to-PCR extraction kits, we found that PicoPure extraction worked the best for our lab when combined with Global Filer and 3500 CE, and they were analyzed through OSIRIS. Now, these samples were epithelial cells, saliva samples that were manually picked. Now, the problem we run into, we cannot manually pick the white blood cells and sperm cells. So we had to integrate into using a semi-automated method. So we use DePere that helps us classify 3 different cell types that are usually encountered at crime scene, which consists of epithelial cells, leukocytes, and sperm cells. So now the next step again is to make a method that is compatible using DePere for the remaining cell types. So we published a paper as well on using DePere to check the compatibility with the reagents through the process. Now, while using DePere, PBS was a vital component in our process because PBS helps to keep the cell viable because we need the cell intact before it goes into extraction. Now, PBS has a tendency to affect PCR amplification negatively. So if it's too high, the PCR efficiency goes down. And we obviously ran into that problem because that initial data showed that with the manufacturer's recommendation of 1x PBS, we saw a dip in our EPG qualities. So then the next step was to find a concentration for PBS that was compatible. for our pipeline. So we looked at 0.5x PBS, 0.25x PBS, and we also decreased the pro-K because we don't work with bulk samples, we work with just one cell, single cell pipeline. And through our data for the samples that were collected to DEB-array, we found that, again, looking at the 3 different matrices, which is average peak height, frequency of alleles detected, and EPG sloping, which is the beta, we saw that PP3 and PP4, which was 0.5x PBS on 0.25 PBS, showed promising results, had a really high median values. Now, there's a reason why we didn't move forward with PP4 instead of it being 0.25x PBS, and that's my next slide. So we looked at the percentage of alleles detected per cell, and the one thing I would like to really stress here is when we looked at PP3 and PP4 closely, 8% of the time in PP3, which was 0.5x PBS, we saw only 0% profile non-detections. No profile was detected. But when we moved on to 0.25x PBS, we see 20% of the time we see no profiles. So even having EPGs with no data is also not good for our pipeline. So that's why keeping everything into consideration, 0.5x PBS with lower pro-K worked the best for us in our lab with global filer and integration with 3500 and OSIRIS analysis. Now, moving forward, the next step was to look at the signal quality that was established to this pipeline. Now, this is just a test set that I'm showcasing right now because our next steps after looking at the signal quality is to use this data. So we have on the x-axis here is 3 different cell types commonly encountered at crime scene, and the y-axis for my first graph is the total intensity, is the total RFU value that we get from each cell after it goes through the process that I just mentioned. We also incorporated sperm cells for our data analysis and to use them in the future for interpretation and clustering, which I'll talk about in the future. But for this talk purposes and where we are at in the project right now, we focus on leukocytes and epithelial cells. So we can see here that for leukocytes and epithelial cells, we see a relatively high median values of RFU, total intensity. For leukocytes, it's around 30,000. For epithelial cells, it's around 24, indicating good total intensity. And then we move into looking at the beta. So the negative, the beta, means that there is sloping. There is some loss of information, lower peak heights with increase in the molecular weight. That is indicative with the little dip over here that we see with the epithelial cells, which could be due to multiple reasons. Maybe the DNA was damaged, there was some problem with the cells. It's not definitive. But at least it shows a correlation that the decrease in the beta is indicated with the lower peak heights indicated here in total intensity. The sperms are haploids, half cells, would have much lower intensity compared to epithelials and leukocytes, which can be seen here as well. We have not integrated sperm cells yet in our future interpretation methods yet, but that's obviously the next steps forward. Now, with all the data we have collected, with everything I showed you with the process, what are the next steps? How do we use single cell is the overarching aim of my presentation, my PhD, and our lab. So moving on is to cluster the single cells and to find a way to get to interpret this data. So moving back to my example of having an admixture from the crime scene. So what happens in single cell? Again, like I mentioned, you would have each tube containing one cell, and then you have one EPG corresponding to that cell. Now, the next step is to cluster, to group this data based on the similarity of the EPG qualities. So we don't just stop by just clustering the data, we look at the clustering performance. So that's the next step. So when we start clustering and grouping the cells, we run into 2 main problems. The positive thing is the correct clustering. Sorry. Okay. So what you want is a correct cluster where each circle indicates one contributor, and the dots and the triangles indicate the different genotypes. So if there are 2 contributors, you would expect all the circles to be together for the 1 contributor and all the triangles to be together for the 2nd contributor. So that's a correct cluster. But for overcluster, what happens is you would see another group of cluster which would have cells from the first contributor being clustered into the other, which is not that bad. It's still a different, it's still just another cluster, but has the same genotypes. But the most catastrophic is the miscluster, where you have a group of cell, where you have a cluster which contains cells from 2 different contributors. So you don't want that. You don't want to have a cluster which contains multiple contributors or more than 2 contributors. So that was the first step to evaluate when we are employing these clustering strategies to look at how many times do we see over, misclustered, or collect clustering. Then the next step is for us to determine the likelihood ratios per cluster. We don't stop there. That's not our final aim, because getting likelihood ratios per cluster is not that viable to us as a community, because we don't work with one cell, we work with an entire admixture. So the final aim is to calculate the likelihood ratio, the average log likelihood ratio for the entire cluster in question. And that assigns the weight of evidence for us. So now let me walk you through the first few steps, which is the clustering performance and calculating the log likelihood ratio for each cluster. And then finally, I will talk about the average log likelihood ratios for the samples that we worked with. So for clustering, we looked at a few different clustering methods, but for the simplicity of the talk and for the time constraints, I'm just going to go over the major 3. One is model-based clustering, then we looked at probabilistic clustering, and then k-mean clustering. So our collaborators helped us look at different clustering methods with the data that we provided through the lab for single cell. So over here, the 2, 3, 4, 5 is the number of contributors. So 2 contributor up to 5 contributor mixtures. The L and H indicates the proportion of the smallest contributor in that mixture. And the y-axis is the proportion, is the percentage of time you see a correct cluster, percentage of time you see over, and a miscluster. For model-based clustering, even after increasing the number of contributors from 2 to 5, we don't see any overclusters or misclusters. So that's promising. For probabilistic clustering, with increasing the number of contributors, we see more of the overclustering. Especially with the 4 and the 5, we see increase in the numbers of lower clusters. Versus the k-means, that would increase in the number of contributors. Not only do we see increased overclusters, but we also see misclusters. So moving forward, we decided to work with model-based clustering for other cell types. This data is only for leukocytes, but we also looked at epithelial cells and leukocytes, which I'll show you in my next slide. So this is what the data looks like for the sample set that I showed you initially, the single cell quality data that I showed in my few slides prior. So same for epithelial cells, we did the same thing across the number of contributors, 2 to 5, and again, L and H indicates the proportion of the smallest contributor. Now the important getaway from these graphs are that for leukocytes, like I said, there is no overclusters but increase in the contributors and no misclusters. But with the epithelial cells, we do see a few overclusters, but that also could be considering the low quality of the data that I showcased in my previous slides, that we had a little lower intensity in the peakites, we had little negative betas, which could be affecting our overclusters in the epithelial cells. Mixed samples are a combination of the epithelials and leukocytes together across different contributor mixtures. And we see that even with increase in the number of contributors for the mixed samples, which consist of good quality leukocytes and average quality of epithelial cells, we don't see any misclusters. So model-based clustering showed us promising results, at least for the sample set that we worked with. So the next step, like I mentioned, is to compute the likelihood ratios per cluster. Thanks to our collaborators, Dr. Lunn helped us develop a software called EZIT. It's evidentiary evaluation of single cells to determine the likelihood ratios per cluster. I won't go much into the details as to what the software does, but this is what it looks like. And let me share you the data for each cluster. So the takeaway, again, the y-axis here is the log likelihood ratio per cluster and across different number of contributors. And I would really like to emphasize on the epithelial cell samples that even when these cells had lower quality EPGs, their log likelihood ratio on an average is the highest density of the log LR falls between 25 to 30, which is really high, even when we are working with low quality samples. And for mixed cells as well, we see at least 25 log likelihood ratio per cluster, which is a good indication that when they're compared against a true contributor, you get a relatively high log likelihood ratios. Now, the final slide is the average per run. So we average per cluster the log likelihood ratio, and this is what we get. And you can see here that regardless of the complexity of the mixture, or if it was a correct cluster, over cluster, miscluster, the highest density of the log LR average falls between 25 to 30 RFU. Even for the misclusters, you see them in that range as well. So even with degraded samples or mixed samples or even misclusters, we get a relatively high log likelihood ratio. So when this is compared with the bulk samples that we have, and we all would know that with the number of contributors increasing for a bulk sample that is run through a traditional pipeline, the log likelihood number dips down. It goes close to zero. And over here you can see that it's relatively high. Like I said, the density falls between 25 to 30 log LR average, which is a good indication to move forward. And lastly, I would like to thank NIJ for the grant. It would not have been possible without the grant funding. And special thanks to my collaborators, my PI, Dr. Catherine Gorgachuk, Dr. Desmond Lunn, and Dr. Ken Duffy for all the help in the pipeline process and all the students who have worked tirelessly on this project. Thank you, and I'm happy to take questions.
Just a quick one. I know you said 30 cycles on the global filer. I'm more familiar with the fusion. Is that increased cycle number, or is that manufacturer recommendations?
No, so we did a study prior in our lab where we looked at as to what's the optimal number of cycles would be for a single copy, and for us it was 30 cycles. So that's what we worked with. So for us, the limit of detection for one copy was 30 cycles for global filer.
Is that above the manufacturer recommendation?
Correct.
Oh, okay.
To my understanding, yes.
All right, so I know that it's 5 o'clock and you think your day is over, but I would like to encourage you to go next door for the poster session between 5 and 6:30. So join us next door for posters.